Welcome to the first chapter of Anatomy Physiology 1. I put this up because a lot of kids say this is the way I explain anatomy. And the reason I do this is if you just have a list of things and you memorize them, like this, very linear, here's one topic, memorize, then a week later, 2 weeks later, it's gone. You don't have it, it didn't stay with you. But if you weave a story around the topics, then when you're trying to remember something, you say, oh, I remember that story. I remember how that works. So oddly enough, in order to remember something, you need to attach it to something. So if you get frustrated with me and think that I'm wandering off the topic, remember that there's method to my madness, and I've been doing this many, many years, and this seems to work the best of all. So that being said, let's get into this topic and see what we're talking about. So our first thing is you're taking anatomy physiology. Well, what does that even mean? Well, anatomy is all the structures that make up your body, and physiology is how they work. How do you use your muscles to move around? How do you use your brain to think? What makes nerve endings fire? That sort of stuff. So that's what we're going to be talking about. And this is human anatomy physiology. So you can compare animals to humans and see how we're alike and how we're different, but that's a whole different course. This is strictly going to be just anatomy physiology, and it's going to be for people who are going into medical fields. So that's what this is for. All right. So how can you look at the structure of the human body? Well, if it's the outside of the body, you can just inspect it. You can look at the eyes, look inside the ears, things like that. So you can actually see certain things without having to hurt the person or the person be dead. Then palpation is where you're feeling. You just like if there's a little fatty tumor, you try to figure out if it's a cyst or a fatty tumor. Is there fluid inside of it, or is it an infection? Is it a solid mass that might be cancer? So palpation or feeling things is a very, it's an art form. It's not completely scientific. So some people are better at it than others, just like some people can draw pictures better than others. Some doctors are able to palpate and be able to feel. Also, physical therapists do a lot of palpation, trying to find where a muscle is out of place or something like that. And then auscultation is trying to listen and hear something. For example, you should have normal bowel sounds. You should have gurgling of food and fecal material moving through the intestines. And if you're blocked, then you're not going to be able to hear that. So I found this little cartoon I thought was kind of funny. I like Gene Wilder. And so when you're trying to auscultate, but they won't stop talking, you're trying to listen to the heart. Another thing you can do is percussion. So if you literally tap on someone, you can hear if there's any air or fluid in an area. They have different sounds. So palpation is more of a rubbing, this is more of a tapping, and this is more of a listening. And then, of course, this is using your eyes. Now, if you're not limited by that, if the person's dead, then you can have a dissection, and you can do gross dissection, and then you can take samples, and then you can look at them under the microscope, so you can really get down to the minutia of the anatomy of people. Another thing that you could do, what they used to do, was they would compare, they would open a pig and say, well, how does that compare to a human? So it was frowned on to do dissection, and it was definitely frowned on to do vivisection, where you cut someone open while they're alive. So there were a lot of things that we couldn't learn, that we had to try to look at animals and guess that that might be what's going on in us also. And that was actually surprisingly successful, but obviously there's a lot of stuff about us that's a little bit different than other animals. We've gotten a lot fancier in the last millennium or so, and we now can look inside the body using x-rays, CAT scans, MRIs. So there's a variety of things that we have, and we'll spend a little time talking about them. So some of you are going to go into some sort of medical imaging, you're going to go into radiology, and so you're going to be helping do x-rays or some of this other visualization of things that are inside the body. Some of you may end up in a laboratory doing histology, looking at microscopic slides to see if you see any cancers, if you see any abnormal structures. So this is a really interesting field of histology. It's very, very necessary, but if you can't sit for a long period of time and you can't stare through a microscope for a long period of time, and that is not a job that you would enjoy, believe me. Cytology, you're looking at cells. So one of the cool things that we've learned to do is to take cells. So we can take a biopsy and we can actually feed the cells, grow them in a petri dish, and then you can perform experiments on them. So I don't have to take a person and cut their kidney out. I can just take a little sample of their kidney and grow those cells up and do all sorts of neat studies with that. So cytology is just a study of individual cells. Histology is a bunch of cells making up a tissue. So there's a little different there. But most of these are going to require the microscope. And then the electron microscope is seriously awesome. They have the scanning electron microscope where you can look at the surface of something, and then they have transmission electron microscope, or TEM for short. And this one, you can actually cut open a cell and look inside of it, or you can cut little things open and look inside of them. So it's very interesting. When you have a free moment, go out and Google or use whatever search engine you use and look at electron microscopic pictures or electron micrographs. And here's one I just went out and found. Electron microscopes freaking people out since 1931. So instead of using light to focus and bounce back off of an object, you use electrons. So you can get much higher magnifications than you can with regular light microscopes. So you can get a decent light microscope for anywhere from $100 to a couple $100. But electron microscopes are going to be more like $500,000 to over $1 million, depending on how fancy you get. So that's not something that you're going to find in most teaching curricula. Usually it's just reserved for graduate students. So now then we talk about physiology, or the function of all those cells, the function of all those tissues that you look at. And some of the subdisciplines that you can go into physiology, you can go into neurophysiology. So you're only just interested in the nervous system and what it's doing, or hormones, or pathophysiology. You're only interested in tissues or cells that are doing something that they're not supposed to. Then, of course, there's comparative physiology, where you look at different species. But again, that's out of the realm of what we do. But a lot of the drugs that we have, a lot of the medical procedures that we have, started out in animals. So they tested them out in animals, and then once they thought they had a handle on it, then they tried it out on humans. So we do need to know a little bit about how closely related certain animals are to us. No matter what medical field that you go into, you're going to have a lot of other things that you're going to have to study in addition to anatomy, physiology, and things like that. So here's somebody who is in the radiology field, and they're talking to somebody and said, oh yeah, my job is easy. You have to know anatomy, radiographic positioning, you have to know about physics, how to properly apply the inverse square law to the kilovolt peak in milliamp per second. So just be aware that anatomy physiology will get you into a program, but you're still going to have to take a lot of other classes in order to get a full picture of what's going on. In this introductory chapter, they spend a lot of time talking about the pioneers in the field, the people who discovered the microscopes, the people who discovered cells, and things of that nature. So I'll talk about things like that, and I will mention some dates, but I'm not really wanting you to memorize the dates so much as to realize that some of the stuff we have known for many thousands of years, and some of the stuff we're just discovering in the last few decades. So this is a constantly evolving, constantly changing field. We're constantly learning new stuff. So it's really neat. Back a long time ago, we believed in things like superstition. We had voodoo. If you kill a chicken, then sometimes it will cure the person. So there were a lot of unusual ideas that didn't always help the patient. And one of the ones that was really prevalent a few hundred years ago was to bleed a person. They thought, well, if the person has evil spirits or evil humors inside of them, if you cut them, then those evil spirits will leave. Well, what they were doing was they were taking somebody who was sick and who was weak, and they would bleed them, and as they bled them, their white blood cells were leaving, their red blood cells that could carry oxygen were leaving, and so the person got very weak. So a lot of people ended up dying from the treatment, whereas if they just left them alone, they might have gotten well. Well, it's kind of interesting because now we laugh and say, ha ha ha, they used to bleed people. Well, one of the ways you could bleed people besides cutting them is you could put leeches on them and let the leeches suck the blood out. And we've come full circle, and now we use leeches again. So if you have a blood clot under your skin, you can put a leech on it, and it will suck up the blood clot through your skin. If you have to reattach a finger, you put leeches on it, and they'll start sucking, and they'll pull the blood through the finger, and they'll eat any of the blood that's clotted or has been in the wound too long. And so they restore blood circulation. So it's kind of interesting how we're going back and looking at old treatments and finding that they actually, some of them worked. The other thing is, whoever was the loudest, the most important, the most listened to, or authoritarian, was the person that they believed. So one of the people, Aristotle, a lot of his ideas were wrong. He was great. He got people thinking. He got people learning. But unfortunately, they said, well, if Aristotle said it, it must be true. And so it took a long time to have somebody brave enough to say, well, that's not actually true. And Sir Isaac Newton, he had all kinds of proclamations and things like that. And we're still believing a lot of the stuff that he said hundreds of years after he's been gone. So if you are an authoritarian figure, if people believe you, you can actually get people to believe stuff that's not true. It's not based on science. So keep that in mind, especially in this day and age of internet where anybody can publish anything about anything. All right, so again, we're going to talk about some of the main people that started bringing science into anatomy physiology, and we stopped listening to people who were important or following superstitious things. I'm sure everybody's heard of the Hippocratic Oath that doctors take, and first do no evil, first do no harm. So he developed a code of ethics, but what is really cool is he said, let's look for natural causes of disease. Let's don't think that gods and demons are what's wrong with the person. So that was pretty good. Aristotle believed that diseases were supernatural, and or some things he thought there was a physical cause. So because he was all over the place, again, he kept us from it going forward for a while until we sorted out what he said that was right and what he said that was not right. One of the things that he did believe in, and we found to be true, is he thinks that complex structures are built from simpler parts. And this is true. Sometimes we go all the way down inside the cell. Sometimes we go all the way into the mitochondria to find out what a problem is. So you may have a whole constellation of symptoms, and you have to drill down and look, not physically drill down, but you look and see the blood work, you look and see how the person functions, you give them tests to see how their heart is working, do visualizations through PET scans or MRIs. So he believed this to be true, and we have found this to be true. So if you can break it down and figure out where along the line the person is having a problem, then you can fix it. Or at least we hope you can fix it. Some things aren't fixable, unfortunately. So Galen has been given a lot of kudos, but unfortunately he was so good that people believed him for almost 500 years before they realized a lot of the stuff that he said was wrong. First of all, he wasn't allowed to look inside of humans because that was banned. So he did animal dissection. He made pronouncements based on animal dissections. But he looked at science as a method of discovery. So this is one of the good things. Instead of just guessing or saying, this is how I feel about something, he actually started saying, let's do, scientific method. Let's do a little bit of actual science before we make pronouncements. But his teachings were adopted all through the Middle Ages. So what he said was what was taught. I don't know if you guys noticed, but the textbook that you're using is written by Saladin. And I have a feeling that this is why this particular person was added to influential people who brought us out of the dark ages of medicine and into modern medicine. So here is one that was a physician to the Egyptian Sultan Saladin. Here's another one who took Galen's findings and Aristotle's findings and combined them with original discoveries and made the canon of medicine. So again, we're getting better, but we're not anywhere near where we need to be because there's so much, and we're still discovering so much. By around 1540s, we had the Catholic Church giving permission to people to do dissection of actual cadavers. So if they're already dead, you could take the dead body and cut it up and look and see. So you can't see how the heart beats, you can't see how the blood flows, but at least you can see the structures that are inside of people. William Harvey wrote several books, and he is kind of the father of experimental physiology. So he did experiments on living organisms. He's the first to realize that blood comes out of the heart and then comes back to it. So one of the things that tickles me in some of the textbooks I've looked at, it shows the blood going out of the heart and coming back. And it forgets to send the blood over to the lungs to get oxygenated. So when we get into the chapter on how the heart circulates blood, we'll show you how it leaves the heart, goes over to the lungs, picks up oxygen, comes back to the heart, and then is squirted out through the aorta throughout the whole body. And then it comes back to the heart, sent back over to the lungs, picks up oxygen, and then sent out to the body again. So he was one of the first to realize that. And if you look, 1628, that's only about 400 years ago that we actually realized how the heart works. And now they teach it in like second grade. Most people know about Robert Hooke because he figured out a way to magnify, and he got all the way up to 30 times magnification, which was amazing for that time. Nowadays, when you're in the laboratory and you're playing with the microscopes, you go anywhere from 40 magnification all the way up to 1,000 magnification with your light microscope. So he only went to 30x, and that was less than what we do with our simplest objective on our microscope. He's the guy who got credit for naming cells because what he looked at under the microscope, he said looked like monk's cells. So this was his microscope. So we actually have his microscope that he used, and this is what he saw. So if you take a slice of tissue, it does kind of look like little cells, little boxes. So that's why we named them cells. Then there was a guy named Hooke, and he discovered a way of magnifying up to 200 times. He did it because he wanted to look at fabric, but he also thought, well, let me look at some other stuff. So he scraped stuff off of his teeth, looked at his sperm, went out and got water from a lake and looked to see what was in it. And of course, you always have to look at blood because you're always, cutting yourself and say, hey, why don't we just look at that in the microscope too? I think it's interesting that Robert Hooke is the guy who gets credit for naming cells, and Hooke is a guy who expanded our use of microscopes. So they're both kind of hooks in their name. So in this introductory chapter, we're going to hit a couple of really big concepts, and one of them is called the cell theory. The cell theory says that all living things can be broken down into cells. Some things are only one cell big, and then others have multiple cells, and then when you get to humans, we've got trillions of cells, and there are so many different kinds of cells in our body. So when we get to the chapter on histology, we'll see if we can make sense of it and kind of group them into types of cells. So we'll talk about that. But they think this was the most important breakthrough in biomedical history to figure out that all living things are made of cells. So that's one of the big concepts you need to learn in this chapter. Another one, oddly enough, is gradients. So we'll talk about gradients in just a second. When you're making pronouncements, when you're doing science and you're coming up with discoveries, you can either do inductive reasoning or you can do deductive reasoning. The most famous person that you probably can think of that did deductive reasoning was Sherlock Holmes. He would look at things and he would say, oh, this is what you ate, you're married, you live in this place, and all this. And people are like, how in the world were you able to deduce all of that? And he would break it down and say, well, I figured it out from this and this and this. So we're going to talk a little bit about inductive reasoning and about deductive reasoning. And we're going to spend a little minute talking about the scientific method. So the scientific method is you look around and you see something and you say, I think that this is a thing. Like, everyone seems to wear red on Monday. So in order to see if that's true or not, you make up a theory. Okay, I believe that if I count so many people on a Monday, then there will be more people wearing red on Monday than on any other day of the week. So you make a hypothesis. If I do this, then this will happen. And then you actually you design the experiment, and you have to make sure that you're not biased, so that you only count people with red, or you count anybody that's got a little bit of pink or a little bit of purple as red because, hey, that's kind of sort of like red. So you have to be careful that you don't influence the results. And then once you've done this, you tally up, you do statistics, and then you come out with your conclusion based on your results. And you say, no, actually people wear red pretty much any day. The only time you're going to find more red is around Christmas. So that would be the scientific method. Some of the people who were proponents of doing scientific method to base it on facts and observations rather than just this is how I feel about something. So Francis Bacon was doing this in England. Descartes in France was doing it. And because these people were so interested in doing scientific discovery and spreading it around and talking, they actually made academies. And you had to be somebody to get invited to the scientific academy. But they would sit around and each of them would present and say, well, this is what I'm working on and this is what I've discovered. And then people were like, oh, that's pretty cool. And they would suggest, why don't you do this? Why don't you do that? So they still have these academies of science in England and France today, where people get together and share their knowledge. So you have to be really careful about inductive reasoning, because here's one of them. You've probably heard stuff like this. All right, penguins are black and white, and some of the old TV shows are in black and white. Therefore, some penguins are old TV shows. So it says, logic is another thing that penguins aren't very good at. So the inductive method is you keep making observations and you keep making observations until you feel like you can draw a generalization. You can say, okay, the sun always comes up in the east. I have observed it every day of my life. It has always come up in the east. It has never come up in the west. And therefore, I feel that I am safe in saying that that's what happens. And I predict that tomorrow the sun will come up in the east also. Another problem you run into when you're making inductive reasonings or even deductive reasonings is, what is your population size? For example, going back to the red. If you did that in America, you might get one set of answers. If you did it in England, you might get a completely different set of answers. If you go to Ireland, famous for wearing of the green, then you're probably not going to be seeing much red at all. And in some cultures, only royalty were allowed to wear purple. So if you did a study on who wears purple, you come up with the conclusion that nobody does. Except for unless you were hanging out and got to see the royalty and saw, oh, well, except for they wear purple. So you do have to be careful about who you look at. Here's an example for you. They came up with the symptoms of a heart attack, and they published the symptoms of a heart attack, and everybody learned them in medical school. But the only people that they looked at were men. So they didn't know what a heart attack looked like in a woman. They just assumed that it was the same. And women have different symptoms when they have a heart attack. So again, you have to be careful about picking your group that you study before you start making pronouncements. So sometimes you tell something that's not exactly true and you didn't mean to, you just didn't think it through all the way. Here's another one of those. I exist and I'm human. My pencil sharpener exists, therefore my pencil sharpener is human. So again, you have to be careful that one thing doesn't necessarily follow the next. If you come up with something that you have found to be true and they publish it and it stands the test of time, if anybody can come along and say, well, but it doesn't work in that particular situation, then you have torn apart the theory. You proved it wrong. So you have to be able to, it has to withstand the test of time, and nobody can retest and come up with something different. An example that comes to mind is the theory of gravity that Isaac Newton came up with. You know, the apple fell down, and he said, well, therefore the earth is attracting the apple, it's pulling it down. And it was believed for hundreds and hundreds of years. And then Einstein came along and said, well, that actually doesn't work. I mean, it works pretty well on Earth, but it doesn't really actually work anywhere else. So that actually, he disproved the theory of gravity. And so they said, well, Einstein, you come up with a theory of gravity. So he came up with one, and then somebody shot down or tore a hole in his theory of gravity. And then we're like, well, okay, we don't have Newton's anymore because it's been disproven, and we don't have Einstein's because his has been disproven. So what are we going to use for a theory of gravity? And so they said, well, let's just go back to using Newton's because it works pretty well on Earth. So even though it doesn't work anywhere else, it's pretty good. Here are some of the things that can mess up your scientific experiment. So if you don't have enough of a sample size, if you don't include men and women, if you don't have people from other cultures, then you're probably not going to have a very good study. And then you have to have controls. You have to have a group of people that you divide it up into two groups, and you treat one of them with whatever it is that you want to prove, and you leave the other group alone and you compare them. So you guys probably heard a lot about this when they were talking about COVID, because when they wanted to test COVID, they needed a group of people and they needed not to treat them. They needed to be the controls, and then they needed to have another group of people who got COVID, and they needed to treat them. And then they could compare. Well, in the group that didn't get any treatment, this many people died. In the group that did get a treatment, this is how many people died. So you run into ethical problems there because you don't want the people in the control group to die. But you need them to die in order to see whether or not the control group and the variable or the treatment group are any different. So that's one of the big problems you run into with experimental designs. And then you have what they call the psychosomatic effects. And this is one of the things when they were first giving people the COVID vaccine, is they would say, we're giving you this vaccine. And some people were just getting a shot of salt water. And some of the people who got the salt water got all kinds of symptoms, And so there's no reason that salt water would give you all those symptoms. So in their mind, they thought that they were going to have ill effects, and so they did. So you always have to look at the psychosomatic effect. And experimenter bias. Here's an example I found of experimental bias. Okay, here is this guy. He goes, the red ones are definitely the tastiest. Have one and see if you agree. So if you couch your question telling the person what answer that you want them to give you, this guy has biased his study. So that one's not going to be any good. And another thing that is important is if you come up with a scientific discovery, you need to run it by people who are authorities in that particular field. So we call this peer-reviewed. Now, used to, if you published something, it had to go through peer review. And certain magazines had higher standards and had more authoritarian peer reviewers, and others were a little less rigorous. So you could try, if you found a really cool discovery, try one of the more rigorous peer-reviewed magazines, and if they turned you down, then you find one that's a little bit looser so that you can get some publications so that you can keep your job. It's called publish or perish. So unfortunately, some people have started publishing things that aren't true, and it's caused all sorts of chaos. One of the biggest ones that comes to mind was a guy who published an article saying that the inoculations that they give little children, when you're born, you start having shots, the DPT shots, and tetanus, diphtheria, pertussis, whooping cough. And he said that that was causing autism. And he published it, and all of a sudden people stopped giving their children the vaccinations. They stopped taking them to the doctor. We saw measles come back. We saw mumps come back. Chickenpox came back. Polio came back. Not nearly at the level it used to be, but we started seeing a resurgence of it. And people looked at his study and realized that it was not a valid study. He just needed to publish something, and he retracted it and admitted, that wasn't a valid. But the damage was done. Somebody had published a paper saying that childhood vaccinations cause autism, and no amount of showing that that is not true has been able to negate that one paper. So it's really important when you publish something that you don't just publish something you made up just in order to keep your job. So keep that in mind. It's kind of scary to think that there's people out there saying stuff that isn't true just to keep their job. Peer review can be absolutely grueling to go through. So you've written this up, you're so proud of yourself, you discovered something, you didn't fudge the data, it's the real deal, and you send it off, and these authoritarian people write on your piece of paper, well, you didn't look at this, you didn't do that, your sample size wasn't big enough, you forgot to include women, you forgot to include kids. So peer review can really tear your paper apart and tear your hypothesis apart, and they make you redo the whole thing or rewrite the whole thing. So I found this little cartoon which I thought was kind of funny. Most scientists regard the new streamlined peer review process as quite an improvement. So all you have to do is dodge physical things instead of verbal attacks on your work. So if you can make it past all these people, then your paper is accepted. So we'll leave theories and laws and hypotheses and scientific method behind us and move on to talking about evolution and why it helps us to understand how humans are formed, what is our anatomy, and what is the function. So we're looking at skulls, we're looking at skeletons. We find people where they've died, and we look around and see what tools they were using, what foods they were eating, where they were living. Were they living in caves? Were they hunter-gatherers? How did they get their food? So we look at things like that. And so in this next section, we're going to talk about evolution, and we're going to talk about natural selection. So Darwin is given credit for the theory of evolution, and He did not say that your great-great-great-grandfather was a monkey. That's not the way that evolution works. But we have found things that swam in the ocean, and then they look very similar to things that a little bit later were found that could walk on land. And we found these things that could walk on land became better predators, faster predators. They fit into the environment better and got warm-blooded so that they were able to hunt both day and night and not just in the day when the sun is shining. So you see all sorts of evolutionary progressions like this when we walked on all fours and then we stood upright. And these I brought up because look how we've evolved. Look how superior we are. Now we're polluting the very waters that we supposedly evolved from. So here were just a couple of cartoons that I thought were kind of interesting, but they show how we think that the changes occurred. So we still have fish. All the fish didn't turn into things that walked on land. All of these things didn't turn into monkeys. All monkeys didn't turn into humans. So evolution is quite a bit misunderstood. And if you want to take, there are whole courses just talking about evolution and looking at the fossil record. It's interesting that Charles Darwin's book was written 150 years ago. He was on a boat and he went to the Galapagos Island and he looked at how things that couldn't interbreed changed over time. So if you look at the turtles on the Galapagos Island, they don't look like the turtles anywhere else you're going to find on Earth. If you looked at the birds and you looked at the beaks of the birds, the finches, that was one of his most famous discoveries, is looking at the beaks of the different finches and how they're different. So he was able to say, some things have changed and some things are isolated and they change in a different direction. They evolve in a different direction. So you learn about geographic isolation, the inability to breed and spread your DNA. If you can't get to the other organisms to have babies by them, then you start having babies that look different. So you diverge, you have divergent evolution. So anyway, that's just some of the things in evolution. All right, and now let's see. Well, let's back up a slide. Okay, so evolution is the change in the genetic composition of a population of organisms. So if you had one person who changed, and we'll just use Spider-Man as a theoretical. So Spider-Man supposedly got bitten by a radioactive spider, and all of a sudden he developed some of the genetic characteristics of a spider, in that he could walk up buildings and he could spin webs through his hands. So he would not be an example of evolution because he's the only one who changed. So in order for evolution to occur, the whole population has to change also. So if you look at what a Neanderthal, it's actually pronounced Neanderthal because there's not the H to go thal. If Neanderthals, look at their skulls, look at our skulls. Look at the way their back is kind of hunched over, and look at the way we're straighter. Our back is straighter than that. So that would be evolution of a whole population. And so that would be considered to be evolution. Now, natural selection is where if you are not the best at surviving in your environment, then you'll die. And if you die, you don't pass on your DNA. So the ones that are left behind are superior in some way. They're faster, they're able to kill more food to provide for their children. So natural selection is whoever's the fastest, the best, whatever, will survive, pass on their genes, and those that aren't as genetically good, I guess you would say, don't get the chance to pass on their genes. So there are selection pressures that they're talking about. If you have predators, so one of the things, I don't know about you guys, but I was raised watching these National Geographic clips, and they always have lions chasing gazelles or chasing some sort of an antelope kind of being, and they kill them and they drag them down and they eat them. And if you look, it's the slower ones, obviously. The faster ones are able to run faster, and so he's getting the ones that are older, sick, or just genetically not up to snuff, not as good. So selection pressures, who gets killed, who lives, that sort of thing. Adaptations. If you happen to be a butterfly and you're born or you emerge from your chrysalis a little bit darker and you get on a building, then the birds will see the other moths or the other butterflies and eat them because they're lighter in color. But because you were just a little bit darker, they didn't see you. So you survive to go on and have more little baby butterflies or more little moths, and yours will be darker yet. And so they're more likely to live. And so eventually you change the color of the moths, or you change the colors of the butterfly to become darker and darker so that they blend in with sooty buildings, because used to they burned coal and they burned wood in order to heat their houses, and so you ended up with a lot of soot that was out in the air and it deposited on the outside of the buildings. So adapting, these are some of the things with natural selection. And then one of the things that, if you've never read about the Darwin Awards, somebody thought that would be a funny idea. And they have, unfortunately, books that they publish of people who have killed themselves doing really stupid things, and it takes them out of the gene pool, so they can't pass on their genes. So here is an example of natural selection. The guy, instead of standing over here and cutting the wood, is standing over here, so once he cuts through the wood, he's going to fall down the mountainside. So they're hilarious. And if they weren't true, and that people ended up dying, but it's astounding what people do